Hello everyone, welcome back to the Petri dish. In today's video, we will understand DNA replication in the easiest possible way using colorful step-by-step poster that I have created. By the end of this video, you will clearly know how the DNA makes its exact copy during the cell cycle. A very important concept for class 10 and class 12 and also for NEET. Before we begin, let us quickly know why the DNA replication is necessary. Okay, so first of all is the DNA molecule doubles. So one of the DNA molecule can have two identical replicas. Okay, one DNA molecule should have two replicas. So that is why the replication occurs. After the cell division, it ensures that both daughter cell must have received one of the DNA from the parent cell. So that is why the DNA replication is important, so that each daughter cell can have one single copy of DNA. That is why DNA replication is essential. Okay, next is we have to understand how the DNA replication proceeds, right? So for the step 1, replication takes place during inside the cell nucleus first of all, and during the interphase. You know the stage also, right? In S phase, right? Let me change the pen. Yeah, in S phase, so the replication occurs. Next is the step 2. Here what happens, the two strands of the DNA helix despiralize or unwind. That means the hydrogen bond between those two strands, they breaks down, okay, and they form a ladder like structure like this. Okay, the weak hydrogen in step 3, the weak hydrogen bonds between the two nitrogen base pairs break down, okay, and they open up, form the replication fork. It is a Y-shaped structure like this. So replication fork is created. Okay, this is the starting point of replication. Next is step 4. Free nucleotides present in the nucleoplasm now come forward and they attach to the exposed bases of the original DNA strand. Okay, cytosine to guanine and adenine to thymine. You need to remember these nitrogen base pairs. So first step, the replication has started during inside the cell nucleus, during the interphase, during the S phase. Next, what has happened? The two strands has unwind and the bonds between them has broken down, and first they form a ladder like structure, and then they have formed this fork, replication fork, from where the replication will begin. Now the new free nucleotides are getting added based on the parent DNA strands. Complementary base pairs will be added like this here, and new strands will be created. How? Based on this, cytosine will bind with guanine and adenine will bind with thymine. Now what will happen? In step 5, as each new nucleotide attaches, it forms a bond with the previous one. Okay, new nucleotide will bond with previous nucleotide by phosphodiester bond. Okay, the joining continues till the complete new strand is already made. Okay, based on the original template strand. Next is each original strand that is there, it acting as a template strand based on which the complementary strand is getting created. Okay, this is why it is semi-conservative model, because the first strand is from the parent generation only and the new strand that is getting formed is only new. So first strand is based on the parent generation only. So that is why DNA replication is semi-conservative, because the new DNA contains one old strand and one new strand. That old strand is called the template strand. Based on that template strand, the complementary strand has been created. That is why it is semi-conservative model. Next is the final and last step. What is happening? DNA replication is completed before the mitosis phase. During the interphase, the doubling of the DNA ensures during the cell division. Each daughter cell, we already discussed that why the DNA replication is happening, because after the interphase, after the cell division, each daughter cell must receive one copy of the DNA. So what is happening during this time? That is why the replication is happening, so that the DNA is being copied and it can be transferred to the new daughter cells that is formed after the cell division process. Okay, So to summarize, DNA replication is beautifully coordinated process involving first the unwinding, then is base pairing, and then building new strands using the old one as template. This ensures the genetic continuity from one cell generation to the next. I hope this explanation has helped you to understand the concept clearly. Don't forget to like, share and subscribe the Petri Dish for more such visual and exam friendly biology content. Thank you for watching everyone.